Morning all of you. Today all of you are welcome in my EEW classroom. I Archana Tiwari going to teach you about lac operon. Now what is lac operon? Let me tell you, it's a very important topic for class 12, and we'll be discussing about this, and in the next class we'll be studying about regulation. First we need to know what is operon, lac operon, and the next class we'll be proceeding with the regulation of lac operon. Well, lac operon, when we use this term lac operon, we can see that two words are occupied here. One is lac, which means lactose, which stands for lactose, and operon is group of genes. Let's write this. Lac stands for lactose, and operon is meant for group of genes. Well, lactose is a disaccharide. Yes, what is this disaccharide? What do you mean by this? Di means two. Yes, disaccharide means a sugar, a carbohydrate which is made up of two simple sugar units, or you can say it's made up of two units generally, which is named here as glucose and galactose. That means lactose is made up of two sugar units. One is glucose and other one is galactose. While glucose is a monosaccharide. So glucose is a monosaccharide which has it's not combined with any sugar units. Lactose is a disaccharide which is made up of two sugar units, and a polysaccharide like starch, dextrose. These are made up of many sugar, small many sugar units. So before we go to this operon concept, let me tell you what is happening there. What is the main funda here? What's the main thing here? The main studying thing here at the back of this concept. Lac operon is available in a prokaryotic cell. In many E. coli cells, you will be finding this operon, where lactose need to be catabolized. Yani ki it needs to be broken down to release energy. You have studied in your earlier classes and lower classes that respiration is a process by which a particular substrate is broken down, generally glucose is broken down to produce energy. Yes. Now, if the glucose is available in the environment where the bacteria survives, it will not go for any other substrate. It will just jump onto glucose and it will start utilizing glucose. ठीक है, अगर glucose मिलेगा तो उसे कुछ नहीं चाहिए. But if glucose is not available in the environment, what will the bacteria do? It will search for another substrate. And if lactose is available in the surrounding environment, it will break down lactose and will produce energy. Now, let's go with the process or rather the structure of lac operon. Let me draw this. This is a structure of operon consisting of three genes: lac Z, lac Y, lac A. Here is the promoter, here is the operator, and in between this there is a part called cap. Here is the lac I gene. So this is a simple operon of the simple lac operon, which I can design to. Now see here, this is the upstream, and this is the downstream. Yes, downstream consists of the structural gene part, and the upstream is the consisting of the regulatory gene part. That means you can say this part is the regulatory part, controlling. This part is the structural gene part. Yes. Now, lac Z, lac Y, lac A. What are these? These are the genes which are generally transcribed and translated to produce three different types of enzymes. Yes. So these three genes are transcribed and then translated, and ultimately three different types of enzymes are produced. Ultimately you can say proteins, which are required for breaking down of lactose to release energy. This is the main theme behind lac operon. Now let's see what lac Z is producing. I said it produces the enzyme beta galactosidase. Y produces permease and A produces transcriptase. acetylase. Now see here. RNA polymerase is an enzyme which is required for the entire process of transcription. This is RNA polymerase. Yes. This RNA polymerase comes and binds to the promoter and the operator site. It just comes and binds and starts transcribing the genes LAC A, LAC Z, LAC Y, LAC A. All the three genes are transcribed, and the process of transcription, what is produced? You know, a single-stranded messenger RNA is produced, and that single-stranded messenger RNA is translated to form these three different types of enzymes. Yes, of which the permease is responsible for increasing the permeability of the lactose. That means it allows the lactose to enter inside the cell from the external medium. It allows its permeability. Understood? And beta galactosidase is responsible for breaking down. Once the lactose enters inside the cell, it reaches inside the cell, it needs to be broken down. Who is responsible for breaking down this lactose? Beta galactosidase. Beta galactosidase breaks down the lactose and ultimately releases the energy. Now the question arises, what is the function of trans-acetylase then? Its function in lac operon is still not known. It still needs to be discovered. But yes, trans-acetylase does one thing. It adds an acetyl-CoA group. Acetyl-CoA group is added from the acetyl-CoA to the beta-galactosidase. This is done by trans-acetylase, which is generally not connected with the lac operon. So you can say the function of trans-acetylase is still not known, but the functions of these two enzymes, as I have said, is done. So ultimately, the main thing is lactose needs to be broken down to release energy. So this is all we have studied in this class, and as I have said in the starting only, regulation of lac operon will be taught in the next class. Thank you, sir.